Okay, so hello everyone. Today's microbiology lectures are going to be about bacterial growth and then bacterial disease. So this is following on from the previous lectures where we looked in detail at bacteria, structure and function. So if you haven't listened to these lectures yet, I'd advise you go back and listen to those before these ones, just because we're going to be using terminology and picking up points that we explained last week. And just again to say, if you've got any issues, are there any topics you'd like to bring up at the revision session, please send me an email at the email address shown on this slide. Okay, so why are we giving you this lecture? Well, as pharmacists, it's really important for you to know how bacteria grow, as you'll need to know this before you can study how drugs such as antibiotics inhibit growth. And you'll be advising patients and doctors on the use of antibiotics, as well as playing a significant role in what we call antimicrobial stewardship. And this is to prevent antibiotic misuse. Okay, so as pharmacists, you'll be heavily involved in this. So today we're going to look at the various factors that affect microbial growth, as well as the different methods of culturing bacteria, including the difference between batch and continuous culture. And we'll also look at the different phases of bacterial growth, and also methods to store bacteria without damaging the cells. And hopefully by the end of the lecture, you'll understand the importance of studying microbial growth. Okay, so first of all, we're going to discuss the different factors which affect bacterial growth. So the growth of bacteria depends on their chemical and physical environment. It's thought that bacteria are present nearly everywhere on Earth, but some bacteria can survive in much harsher environments than others. For example, certain bacteria live in Arctic ice or hot springs. And bacteria that survive in these extreme conditions are called extremophiles. However, most bacterial species wouldn't survive in such harsh environments. In fact, all bacteria do need to respond to the environment, but if the conditions become too unfavourable and they can no longer adapt, then they will stop growing and die. So for optimal growth, bacteria really need to be in the correct environment. So like humans, all bacteria will need specific nutrients to grow, and this will include sources of carbon such as glucose, as well as nitrogen for the synthesis of amino acids for protein synthesis, as well as nitrogen is needed for the synthesis of nucleic acids like DNA. So the specific nutrients required will depend on the bacteria species. So when you have bacteria grown in a lab, for example in hospital or industry, you must make sure they've got all the nutrients they require in the culture media and that they are placed in the correct environment, for example the correct temperature, correct level of oxygen. Just to explain quickly about culture medium, this is a liquid or gel that's designed to support the growth of bacteria, but we're going to cover this more later on about the different types of culture media. And when we're growing bacteria media in the lab, we only actually want the specific bacteria that we're interested in to grow. So we don't want the growth of other microbes. So therefore, we'll put substances in the media that will inhibit the growth of other microbes. For example, antifungals, which will inhibit the growth of fungi. Okay, so the next two slides look at some of the major factors that affect bacterial growth. First factors are solute concentration and water activity, and these relate to the process of osmosis. So when we are talking about solute refers to all the molecules or ions dissolved in water, and the process of osmosis is the diffusion of water across a membrane from a low solute concentration to a high solute concentration. Okay, so that's a low solute concentration to a high solute concentration. So the water will move to equalize the concentrations. And if we take the example of a bacterium in a hypotonic solution, so this is a solution with a lower solute concentration, also called osmotic concentration, than the inside of the cell. Okay, so the solution has a lower solute concentration than the inside of the cell. And if the bacterium is placed in this hypotonic solution, then water will move into the bacterial cell, causing the cell to swell. If you remember from the last lecture, bacteria actually have a cell wall that helps protect the wall from actually rupturing in this situation. Whereas on the other hand, if the bacterium is placed in a hypertonic solution, that's hypertonic solution, so that's a solution with a higher solute concentration than inside the bacterial cell, then in this situation the water will flow out of the cell. And most bacteria actually require an isotonic environment or a hypotonic environment for optimum growth. And when we're talking about an isotonic solution, this is a solution that has the same solute concentration as inside the cell. So under these conditions, there's no net movement of water. The amount of water entering or exiting the membrane is equal. Certain bacteria, on the other hand, are what we call halophiles and require the presence of high salt levels such as sodium chloride to grow. And there are also some bacteria that are osmotolerant, and these bacteria are able to grow over a wide range of osmotic concentrations. And pH is another important factor for growth. So you have acidophiles, bacteria that are acidophiles, and they have their growth optimum between 0 to 5.5 on the pH scale. And then we have neutrophiles, and these have their growth optimum between 5.5 to 8. And then we have the alkalophiles, which have their growth optimum between 8 and 11.5. Okay, so if you remember probably from A level, the pH scale measures the acidity. Okay, so lower on the scale is very acidic, higher on the scale is alkaline. And around pH 7 is neutral. Okay, so most bacteria are actually neutrophiles. So as we mentioned earlier, carbon is really important for bacteria. So it's really important for bacteria to have a carbon source. And this can be from an organic source, such as glucose, or from carbon dioxide. And this depends on the bacteria. And just to tell you that when we say organic compounds, we mean compounds with carbon and hydrogen. Okay, and then the next factor we'll talk about is oxygen. So different bacteria have different relations with oxygen. So obligate aerobes, with these bacteria, they need oxygen for growth. Okay, it's essential. Then we have the facultative anaerobes. Okay, so they grow best in oxygen, but they can grow without oxygen. Then we have the aerotolerant anaerobes, and these can grow equally well with or without oxygen. Then you have the obligate anaerobes, and these cannot grow in the presence of oxygen, so oxygen is toxic to them. And then lastly, we've got the microaerophiles. Okay, and they require a low level of oxygen, so too much oxygen is toxic to these bacteria. Okay, so external temperature can have an important effect on bacteria as they are unable to regulate their internal temperature, unlike humans. So changes in external temperature will therefore affect bacterial enzyme activity. So enzymes will work at an optimum temperature, and if you alter the temperature, then they won't work as well, and then this will affect the growth. So bacteria therefore have a minimum, optimum and maximum temperature for growth and can be divided into groups depending on their optimum growth temperature. So first of all, you have the psychrophiles, and these are what's known as the cold-loving bacteria, and these are usually found in the Arctic or Antarctic. Then you have the mesophiles, and these are bacteria that grow best at moderate temperatures, and most bacteria fit into this category. Then you have the thermophiles, and as the name suggests, these are the heat-loving bacteria, and these are commonly found in hot springs. Then you have the hyperthermophiles, and these can grow at extremely high temperatures. For example, bacteria which live in the Atlantic Ocean hydrothermal vents. Okay, so these are like hot springs on the ocean floor. So in terms of pressure, there are certain bacteria for which pressure is an important factor. For bacteria which live on the land and the surface of the water, they're not really affected by pressure. But for bacteria living, for example, at the bottom of the ocean, they need to be able to withstand high hydrostatic pressure. So bacteria that can grow and reproduce at high pressures are barotolerant. And then you've also got barophilic bacteria, and these actually require the high pressures to grow. Okay, and next we'll look at methods of bacterial growth. So we talked about binary fission in the last lecture. And if you remember, this is asexual reproduction of bacteria. And this is where the parent bacteria cell divides into two identical daughter cells. And this is the most common form of bacterial reproduction. Some bacteria have other methods of bacterial growth, for example, budding. So budding is another form of asexual reproduction, but its process is quite different to binary fission. So with budding, the bud first appears as a small projection at a single point on the mother cell. This new cell then pinches free of the mother cell and is noticeably smaller than the parent. Filamentous bacteria, on the other hand, reproduce by breaking into fragments, and then each fragment then grows into a daughter cell. We talked about sporulation in the last lecture, if you remember. So this is a process by which bacteria can form endospores when conditions become unfavourable. And these spores allow the bacteria to lie dormant, and then when the conditions become more favourable, they can become vegetative cells, which are metabolically active again. Okay, so as we touched upon earlier, culture medium is a liquid or solid preparation used to grow bacteria in the lab. So medium is a singular, whereas media is plural. And the most common growth media are nutrient broths and agar plates. So agar plates are solid preparations, most commonly used for small scale culture. You can see the agar plate below on the slide. And these are used for small scale culture, for example, in hospital labs for antimicrobial susceptibility testing. So for antimicrobial susceptibility testing, what happens is they spread the bacteria on an agar plate. So if the person has an infection and they want to know what antibacterial can treat it, okay, they take the sample and they spread the bacteria on the agar plate, and then they place paper discs which are impregnated with antibiotics onto the plate. And then after incubation, growth of the bacteria is observed. And then the areas around the antibiotic discs where no bacterial growth can be seen are what we call zones of inhibition. And these zones show that an antibiotic has been successful in stopping the bacterial growth or killing the bacteria. And you can place several antibiotic discs on the plate, and you can measure and compare the inhibition zones. And this way you can compare the efficacies of different antibiotics. So liquid culture in broths, on the other hand, is useful for mass large-scale culture of bacteria. For example, as we talked about in the last lecture, recombinant bacteria. So liquid broths can be used for the mass culture of recombinant bacteria. For example, bacteria that produce medically important proteins, for example, insulin. Okay, so, and this is a good way to get a large amount of product. Okay, so if you have a sample with a mixture of bacteria, so what we call a mixed population, but you're interested in a single bacterial species, you will need to obtain what's called a pure culture. And this is a population of cells arising from a single cell. And one way you can do this is by the streak plate method. And the first step of the streak plate method is to add the inoculum to the edge of the agar plate. Okay, and by inoculum, we mean the sample of bacterial cells added to start the culture. The inoculum is then diluted by streaking it across the surface of the agar plate. And then while streaking successive areas of the plate, the inoculum is then diluted to the point where there is only one bacterial cell deposited every few millimeters on the surface of the agar plate. So then when these lone bacterial cells divide, they will form a cluster, what we call a colony. And these well-isolated colonies can then be picked out and restreaked on a fresh agar plate to produce pure culture. So another method of isolating a pure culture from a mixed population of bacteria is the spread plate method. So in the spread plate method, a liquid sample of diluted bacteria is transferred to the center of an agar plate and spread evenly over the surface with a sterile bent rod. The dispersed bacterial cells then develop into isolated colonies, which can then be counted. Okay, so next we'll look at bacterial growth in liquid culture or broth, as shown in the pictures here. So as we said, liquid culture is usually used for large-scale culture of bacteria, for example, in industry. So cultures in liquid media can be batch or continuous. So in batch culture, bacteria are incubated in a closed culture vessel, like a test tube or a flask, with a single batch of medium. So fresh medium is not provided during incubation. And what this means is, as the nutrients are used up, they're not replaced, so their concentrations decrease. And waste products also accumulate because they're not removed. So this means that only a few generations of bacteria are possible. There are a number of advantages to this type of system. For example, it's versatile, so it can be used for different reactions every day. It's safe as it can be properly sterilized, and there's little risk of infection or strain mutation. However, there's disadvantages such as high labour cost, idle time, and there can be problems with safety when filling and emptying and cleaning. So continuous culture, however, in contrast to batch culture, maintains a continuous supply of nutrients and removal of waste products. So therefore several generations of bacteria are possible. And one of the main types of continuous culture systems is the chemostat. And a chemostat is designed so that the rate at which the medium is pumped in is the same as the rate at which the medium containing bacteria is removed. So there are several advantages to continuous culture. So works all the time, there's low labour cost and good utilisation of the reactor. It's also usually efficient, and also you can automate, and the product quality is constant with this. The disadvantages, however, are you can get infections and spontaneous mutations. So in this case, the continuous production fails. The setup is also inflexible, so it can really be used for other productions without undergoing substantial modification.